This required practical investigates the effects of osmosis on plant tissue, and by the end of this you should be able to describe how to investigate the effects of concentration of solution on the rate of osmosis, how to calculate percentage mass change, and how to identify the concentration of cell sap inside the plant cells. Firstly, osmosis is the diffusion of water from a dilute solution to a concentrated solution through a partially permeable membrane. A dilute solution is one where you have a high concentration of water molecules compared to a low concentration of dissolved solute. That could be salt or sugar. A concentrated solution is the opposite. This is where you have a low concentration of water molecules compared to the concentration of solute. When cells are placed in a dilute solution, water will move into the plant cells because there's a higher concentration of water in the beaker. In cells that are placed in a concentrated solution, water will move out of the cells because there's a higher concentration of water inside the cells than inside the beaker. In this investigation, the independent variable is concentration. It's always the subject of the investigation. Sometimes they could ask about the effect of temperature on the rate of osmosis, and in that case temperature would be the independent variable, and also they could give you an investigation investigating the effect of surface area, and again that would be the independent variable. The dependent variable is the variable that you measure, and in this case we are going to measure the initial mass and the final mass in order then to work out the percentage change in mass. The control variables are the things that you keep the same to make it reproducible. When they give you a diagram like they have in this question, it's a gift because you can see immediately things that they keep the same. For example, they all have used the same volume of solution. Secondly, you can see they've all used the same size of plant cell. It could be length or it could be mass. You may have to stipulate. Equally, they will have conducted the investigation at the same time, so therefore it would have been the same temperature, and you would have left the cells in the solution for the same length of time. You need to be able to calculate percentage gain or loss of mass in cells. The formula for this is the change in mass divided by the total mass times by 100. The change in mass is calculated by subtracting the initial mass from the final mass. In Beaker 2, we can use the data from the table at the top to work out the change in mass, and then we can use that figure to calculate the percentage change. So change in mass is 12.2 minus 10.6. That gives us a value of 1.6. We would use the change in mass then divided by the total mass at the beginning, which is 10.6, times by 100. You can use your calculator and you would get a value of 15%, and it's a positive number, so therefore it indicates that it gains mass. In Beaker 4, on the other hand, again we'd use the data at the top of the table, the final mass and the initial mass, so it would be 7.9 minus 10.1. That gives us a value of minus 2.2, so that's the change in mass. We put that then into our percentage mass formula, so it would be minus 2.2 divided by 10.1 times by 100, and that would give us a value of minus 21.75. The data in the table is only to one decimal place, so we would then round that up to minus 21.8, and that is a loss. If you are asked to plot the data, then it's important that you read the question carefully. It generally asks you to do at least 2 or 3 things. In this question, it asks you to choose a suitable scale and label for the y-axis, plot the results and draw a line of best fit. As you can see, I've gone ahead and written in the scale for the y-axis. Once you've done that, you need to work out then what one little square on the graph paper is equal to. To do this, pick 2 points on your y-axis and count how many little squares are between it. So for example, between 0 and 5 there are 10 little squares. We would then do 5 divided by 10, which equals 0.5. When you're plotting your data, it helps you to be more accurate because you can work out then where numbers such as 12 or 24, which are not on a major grid line, are. Once you've plotted your data, then go ahead and draw your line of best fit using a pencil and draw it freehand. Do not use a ruler. Once you've drawn in your line of best fit, you can then work out what the concentration of the cell sap is inside the cells. This is where your line of best fit crosses the x-axis here. And again, you would have to work out what your scale is going across the x-axis. So again, we would pick 2 points on it. So between 0.2 and 0.4, we have 10 little squares. So this time it would be 0.2 divided by 10, and that comes to 0.02. So we can work out precisely then where our line crosses, and we can see at 0.4 it goes one little square over. So therefore our concentration of cell sap inside our cells is 0.44 grams of sugar. In order to explain mass gain or mass loss, you need to be able to include 2 things in your answer. Firstly, the difference in concentration between the cell and the beaker, and then secondly, whether water moved in or out of the cells. Be very careful when you're constructing your answer because you need to make sure that you talk about one or the other. So for example, you either talk about the concentration of water in the cell compared to the concentration of water in the beaker, or you could talk about the concentration of solute inside the cell compared to the concentration of solute in the beaker. In the model answer that I've written on the PowerPoint, I've talked specifically about the concentration of water, and that's in both where they gained mass and where they lost mass. Before you go into the exam, you need to know the difference between eukaryotic and prokaryotic cells. Eukaryotic cells include all plant and animal cells and are therefore multicellular organisms. Prokaryotic cells are bacteria and are single-celled organisms. So immediately you should be able to know with a question like this that the 2 cells on the right, the liver cell and the mesophyll cell, are plant and animal cells and are therefore eukaryotic, and the bacterial cell is prokaryotic. This question only asks for 2 similarities between prokaryotic cells and eukaryotic cells. There are 3 differences, so make sure you can recall those 3 differences off by heart. Just by looking at the images, you can see that they all have cytoplasm, they all have a cell membrane, and thirdly, They all have ribosomes, which are indicated by these small black dots in all 3 cells. You also need to know the 4 differences between a prokaryotic and eukaryotic cell. So you can stop the video and you can recap the differences in your own time. This question only asks for 3, but there are 4 differences. When you're writing your answer, think about your technique before you start writing. Are you going to write about how the prokaryotic cell is different from the eukaryotic, or are you going to describe how the eukaryotic is different from the prokaryotic? So make sure you're clear in your answer, like I have been in the model answer on the PowerPoint. Firstly, prokaryotic cells are much smaller, and you can tell this from the diagram. If you look at the bottom of all the images, you can see that the bacterial cell and the liver cell are in micrometers. But then if you look at the mesophyll, it's in millimeters. Millimeters is a bigger unit than a micro unit. And so they've just really put that in there just to test you. Secondly, prokaryotic cells don't have a nucleus, whereas eukaryotic cells do. You can see the nucleus clearly in the 2 eukaryotic cells. Thirdly, in prokaryotic cells, the DNA is free in the cytoplasm, as you can see by this long strand here. And secondly, it also has DNA in plasmids, which are little loops. And then finally, a 4th difference is that prokaryotic cells have no mitochondria. And again, you can see this on the diagrams. Both the mesophyll and the liver cell have mitochondria, which is always represented by this oblong shape with a little wavy line in it. There are 2 types of respiration that you need to be able to describe, explain and compare. Aerobic respiration takes place in living cells and uses oxygen and glucose to produce energy, carbon dioxide and water. This type of respiration produces the most amount of energy, and that's what animals use mostly. Anaerobic respiration in animal cells is generally when the muscles become tired and there's a lack of oxygen. And the word equation for this differs because you've only got glucose before the arrow as the reactant, and then the product is lactic acid. Lactic acid is the chemical that causes cramp in muscles, and this would need to be broken down following exercise when you start to breathe in more oxygen, and that oxygen helps to oxidize the lactic acid. This only produces a small amount of energy, and it's not obviously the preferred method of our cells. Anaerobic respiration in yeast is also known as fermentation, and again this lacks oxygen. It starts off with glucose, and when broken down it produces ethanol and carbon dioxide. Ethanol is used in the brewing industry to make alcohol, and the carbon dioxide that's produced is used to make bread rise. You are quite often asked to compare anaerobic and aerobic respiration. So if you can recall the word equations, you can write them at the top of the page if they aren't given, and this will help you then when you're writing your answer. Compare means you would describe the similarities and you would then describe the differences. Before you write your answer, make sure you think about your technique. Are you going to write it in paragraphs, i.e. the similarities and then a paragraph for the differences, or are you going to write it point for point? I would recommend you write it point for point if you find it hard to write a long extended answer in a paragraph. So like I said, think about first of all your technique, point for point or paragraph, and then secondly, make sure when you are writing out your statements, make sure that you specify which type of respiration you are actually talking about. There are 2 similarities. Both of them use glucose and both of them take place in cells. There are 4 differences that you need to be able to recall. Aerobic respiration uses oxygen And a good connective here would be whereas anaerobic respiration doesn't. Secondly, aerobic respiration is used by eukaryotic cells. However, anaerobic respiration is used by prokaryotics. Thirdly, aerobic respiration releases lots of energy, whereas anaerobic respiration only releases a little bit. And finally, aerobic respiration takes place in the mitochondria, whereas anaerobic respiration takes place in the cytoplasm. This would be the information that I would use on a flashcard if you've got time to make any of them tonight. Finally, I'd just like to say good luck in the biology exam tomorrow morning. Thank you.